Hello. So we have four main topics. You can see here this is a product for protein that is in the market, and you can see how the lab supervisor prepare a purification column to separate a commercial biological product. So for the first topic, subtopic, proteins as a biotechnology product. Proteins is a large molecules, of course, will go fast through that required for structure, function, and regulation of living organisms. In 2000, NIH launched the Protein Structure Initiative, and it's an effort to identify the structure of human proteins. The goal is to able to model unknown protein structure based on structure comparison to those stored in the database. More than 1200 superfamilies of protein are identified so far. The relationship between proteins amino acid sequence to its structure is well understood, and there is a public database hold more than 33,000 protein sequences. The recombinant DNA technology made it possible to produce a specific protein on demand, like enzymes, hormones, and antibodies. And this table is for knowledge. You can see the some enzymes and their industrial applications. This table as well for knowledge, you can see some protein-based pharmaceutical product. Most of them are produced as recombinant proteins. These biotechnology drugs and other medical application produced through microbial fermentation or mammalian cell culture. These can produce large batches of protein products in a bioreactor by growing the host cells that have been transformed to contain the therapeutic gene of interest. The cells are then stimulated to produce the target proteins through precise culture conditions that include balance of temperature, oxygen, acidity, and other variables. At appropriate time, the proteins are isolated from the culture, tested at every step of purification, and formulated into pharmaceutical active product. The second subtopic is protein structure, and of course most of you are familiar with that. You have a primary protein structure that is like B-shaped, like in the first figure, and due to the formation of the bonds between the hydrophobic amino acids and the alpha helix and beta sheets, a secondary structure appear, which is the alpha helix and beta sheet. These alpha helix and beta sheets soon come into three-dimensional polypeptide chain and are formed when these secondary structures are close linked, and these once reaching this level of complexity, the tertiary structure, these determine the protein function. Then you have the quaternary structure, which is unique for each protein, and it's globular, and it's a three-dimensional complex built of several polypeptide, not only one polypeptide. And here you can see a structure of a protein complex that consists of four polypeptides. Protein folding. The structure and function of a protein depends on protein folding. If a protein folded incorrectly, so its function, desired function of protein is lost, and misfolding protein are can be degraded inside the cell. In 1951, the alpha helix and beta sheets were discovered. Their structure are very fragile, and the incorrectly folded protein can lead to disease, including Alzheimer, cystic fibrosis, and mad cow disease, and other forms of cancer, and even some heart diseases. A challenge of biotechnology is to understand and control the protein function folding in the manufacture process. So actually in a biotechnology factory, you can produce the protein, but due to some technical errors, you may not have it properly folded, so no function for that protein. So it must the biotechnology uses that to understand. The process of glycosylation is a very important process for the function of protein. It's a post-translational modification wherein carbohydrate units are attached to specific location on proteins. More than 100 post-translational modifications occur within eukaryotic cells. This change can affect the protein activity by increasing its solubility, orient protein to the membrane, extend the active life of molecule in an organism. This glycoprotein, the spray protein undergo glycosylation, can be used to target and destroy B lymphoma cancer cells. The glycoprotein is combined with a nanoparticle loaded as a chemotherapy drug. Protein engineering by directed molecular evolution. It is the introducing of a specific predefined alteration in the amino acid sequence through a process called directed molecular evolution technology. It can induce mutation randomly into gene and then select the bacteria with the protein product enzyme that have the high activity. That's meaning you do mutation, as we said before, single nucleotide mutation, and you check if this mutation will increase the activity of that protein. Example, a company was able to produce bacteria and the industrial enzyme that tolerate high cyanide concentration that normally kill bacteria. Resulting selected bacteria can be used to remediate cyanide contamination resulting from mining and other waste accumulation. The protein engineering provides the possibility to invent proteins that are tailored or designed for a specific application. Example, using biotechnology to form synthetic infectious protein particle to study the following diseases. The faulty protein folding is expressed as a disease caused by infectious protein called brines, PRPC, which attract normal cell protein and induce a change in their structure, leading to the accumulation of useless protein that actually damages the cell. So brines can occur in sheep and goats and cows and causes mad cow disease. In human, it causes another a similar type of disease named according to the two scientists who discovered Creutzfeldt and Jakob disease, CGD, and this is a fatal degenerative brain disorder. The third subtopic is protein production. There are two major phases used in producing proteins, either upstream processing or downstream processing. Upstream includes the actual expression of a protein in cell, while the downstream involves the purification and verification of the function of the protein. In the upstream, we will talk about first using bacteria in upstream processing. Most common bacteria species used is the E. coli. They cultured in large quantity in a short period of time. They're relatively easy to alter genetically and can increase the level of production of bacterial protein by introducing additional copies of relevant gene. So you can introduce in the bacteria not single copy of a gene, you can introduce a multiple copy of genes, so to make sure to have a multiple copies of protein translated. The majority of proteins synthesized naturally by E. coli are intracellular. Most cases resultant foreign protein accumulate in the cells in the bacterial cytoplasm in the form of insoluble clumps, which are called inclusion bodies, which must be purified from other protein. Sometimes the genetically engineered E. coli produce the desired protein in the form of a fusion protein. That means it's attached to another protein. So the target protein is fused to bacterial protein, so an additional step is necessary to break these two parts. Fused bacterial protein is usually an enzyme that will bind to its substrate and can be attached to a purification column. So you use this step to purify the protein by attaching it by engineering and attaching it to another protein that used to purify. There are cons to expressing proteins in bacteria. Why? Because protein expression upstream. So we're talking about now about the fungi, using the fungi as in upstream processing. So it's we can use that source of a wide range of proteins used in products including animal feed and beer. Many species of fungi are used as a host for engineering products. These fungi are eukaryotic and capable of doing post-translational modification to allow proper folding of the proteins. You can see from this table, it's a knowledge table, some recombinant protein that is being produced from different types of fungi. If we use the plant as an upstream processing, plants can be genetically engineered to produce a specific protein that do not occur naturally. Tobacco plant is a great choice for biotech protein production. The disadvantage using a plant, not all protein can be expressed in plant. They have a cell wall which makes purification more difficult. The process of glycosylation is highly different from animal cell. We'll use mammalian cells as upstream processing. Actually, the nutritional requirement for a mammalian cell is quite complex. The mammalian cell grows very slowly and is very challenging. They can be easily contaminated. It is the best choice for protein designed to be used for humans. If we use animal bioreactor as upstream processing, we can use a monoclonal antibody production. Mice are injected with an antigen and the mouse secrete the desired antibody, and this antibody is then purified. If we use an insect system as upstream processing, the most famous is the baculovirus, which is the virus which infected the insects, are used as a vehicle to insert the mammalian DNA, causing the desired protein to be produced by insect cells. Sometimes the post-translation modification of proteins is slightly different in insects than in mammals. Currently, this technology is used for small quantities of protein which are needed for research.